Hello friends, hi, good morning, welcome to our channel, The Voice of Nurse. Today we are going to discuss continuous topic from the previous videos. The subject name is microbiology. Till now we finished two parts related to the microbiology subject. This is part 3 video related to the microbiology subject from the question number 51 to further question we are going to discuss today. Till now we finished part 1 and part 2 related to the microbiology. In part 1 we discussed related to the microbiology along with introduction, definition of microbiology, what is microorganisms, type of microorganism, what is infection and infection transmission, immunity and types of immunity. Along with these topics plus who is father of microbiology, who is father of antiseptic surgery and sterilization techniques introduced by whom. These all are things we discussed in part 1 video related to the microbiology subject. Along with this we discussed 20 FAQs, very important questions, and FAQs frequently asked questions in previous years exams. And part 2 video we continuously discussed related to the microbiology from 21 question to 50 questions. 30 questions we discussed in part 2 video. I will give a link in description. Please check it once friends. Today we are going to discuss part 3 from the question number 51 to further questions we will discuss today class. Before moving on to topic, a small request friends, anybody watching first time my channel, please subscribe my channel. Click on the bell icon for further notification. Please, if you like a content, give me like and please share your opinion in comment section and share the video to your friends. Let's move on to topic discussion. Today class is part 3 related to the microbiology subject from the question number 51 to further question we are going to discuss now. The 51 question, artificial acquired immunity develops by. The artificial acquired immunity will develops by. We will see here options. Option A, vaccination. Option B, after an attack of infections. Option C, repeated exposure to infection. Option D, naturally. The active artificial acquired immunity refers to any immunization with an antigen. By giving a safe form of the antigen artificially, the body will produce its own antibodies and, more importantly, develop circulating long-lived B memory cells with high affinity B cell receptors on their surface. The active artificial acquired immunity refers any immunization with an antigen, means we have to give a vaccine. So here artificial acquired immunity will develops through the vaccination. So here in these options, option A is the correct answer. Here naturally means we will get at birth only. Naturally means automatically the artificial acquired immunity will develops by the vaccination, but naturally it will occur at birth. So here in these options, artificial acquired immunity develops by vaccination. Option A is the correct answer. Next we will move on to question number 52. Hot air oven works based on the principle of. The hot air oven works based on the which principle? We will see here options. Option A, moist heat. Option B, dry heat. Option C, disinfection. Option D, incineration. We will see one by one. Moist heat. Moist heat sterilization is performed in a pressurized chamber called an autoclave. So autoclave method we will use moist heat. Next option B, dry heat. The proper time and temperature for dry heat sterilization is 160 degrees Celsius for 2 hours or 170 degrees Celsius for 1 hour, and in case high velocity hot air sterilizers in 190 degrees Celsius for 6 to 12 minutes. Item should be dry before the sterilization since water will interference with the process. The hot air oven is a type of dry heat sterilization. So here option B is the correct answer. Hot air oven works based on the principle of dry heat. The dry heat sterilization is used an equipment that cannot be wet and on material that will not melt, change form when exposed to high temperature. So hot air oven works based on the principle of dry heat. Option B is the correct answer. Next we will move on to question number 53. Gram positive bacteria appears in which color? The gram positive bacteria will be appears in which color? We will see here options. Option A green, option B pink, option C yellow, option D violet. On applying a strain, gram positive bacteria appears blue or purple. So here violet is there. Violet is the correct answer here. Under the microscope, on applying a strain, gram positive bacteria appears blue or purple in the color under a microscope. One of the example for the gram positive bacteria is Staphylococcus aureus. In case gram negative organisms are either pink or red in color. So here gram positive means it will be appears in the color of blue or purple. Gram negative organism either pink or red in color. In this question gram positive bacteria will appears in which color means here option D violet color is the correct answer. Next we will move on to question number 54. The term mutation was coined by. The term mutation is coined by which scientist? We will see here first options. Option A Darwin, option B Lamarck, option C Hugo de Vries, option D Pasteur. What is mutation? Mutation means any change in the DNA sequence of a cell is called as mutation. Mutations may be caused by mistakes during cell division or they may be caused by exposure to DNA damaging agents in the environment. Mutations can be harmful, beneficial or have no effects in sometimes. Here in the year of 1901 the scientist Hugo de Vries. So here option C is the correct answer. In the year of 1901, the geneticist Hugo de Vries gave the name mutation to seemingly new forms that suddenly arose in the experiments. And he is the father of mutation theory. Hugo de Vries, he is father of mutation theory also. So here in these options, the term mutation was coined by option C, Hugo de Vries is the correct answer. Next we will move on to question number 55. Mesosomes are the part of. Mesosomes are the part of. We will see here options. Option A lysosome, option B Golgi, option C plasma membrane, option D all of the above. We will see one by one. What is lysosome? First we will see option A lysosome. Lysosomes are membrane enclosed organelles that contain and carry off enzymes capable of breaking down all types of biological polymers like proteins, nucleic acid, carbohydrates and lipids. Here there is no involvement. Mesosomes are part of the lysosomes. Next option B Golgi. Nothing but Golgi bodies. What is Golgi bodies? A stack of small flat sacs formed by membranes inside the cell's cytoplasm is called as Golgi bodies. So next we will see option C plasma membrane. Plasma membrane. What is means by plasma membrane? The plasma membrane is also called as cell membrane. Is the membrane found in all cells that separates the interior of the cell from the outside environment. It will be like a form of mesosomes. The mesosomes are organelles formed by plasma membranes in foldings. They are found in the membranes of bacterial cells. Mesosomes are the areas in the cell's membrane of prokaryotic cell which folds inwards. It helps in cell wall formation, DNA replication, respiration and increases the surface areas of the plasma membrane. So here mesosomes are the part of the plasma membranes. In these options, option C is the correct answer. Next we will move on to question number 56. Typhoid fever involves. The typhoid fever involves, we will see here options. Option A skin, option B muscles, option C nervous system, option D gastrointestinal. The typhoid fever or typhoid is an infection that spreads through contaminated food and water. Typhoid fever, also called as enteric fever. The typhoid fever is caused by Salmonella bacteria. Typhoid fever is rare and in the blood. They travel to lymph nodes, gallbladder, liver, spleen and other parts of the body. So person with typhoid fever carry the bacteria in their blood and bloodstream and intestinal tract. The signs and symptoms for the typhoid fever like high fever, fatigue, headache, nausea, abdominal cramp, abdominal pain, constipation or diarrhea. Some patients have rashes also. So here the typhoid fever is an spreads through the contaminated food and water. So it will be involved in the system is option D, gastrointestinal system is involved. So here typhoid fever involves here option D, gastrointestinal is the correct answer. Next we will move on to question number 57. What is mycelium? What is mycelium? We will see options first. Option A fungus, option B virus, option C protozoa, option D bacteria. A mycelium is a network of fungal threads or hyphae, the mass of branched tubular filaments of fungi. The mycelium makes up the thallus or undifferentiated body of a typical fungus. Mycelium is mainly composed of natural polymerase as chitin, cellulose, proteins and etc. So here what is mycelium means, here option A fungus is the correct answer. Mycelium is a fungal threads or hyphae mass of branched tubular filaments of fungi. So here in these options, option A is the correct answer, fungus. Next we will move on to question number 58. Mumps is viral infection that affects which part of the body? The mumps is a viral infection that affects which part of the body? We will see options. Option A ears, option B parotid glands, option C reproductive organs, option D all of the above. What is mumps? Mumps is also called as parotitis, a viral infection that affects the salivary glands that easily preventable by a vaccine. Mumps affects the parotid glands, salivary glands below and in front of the ears. The disease spreads through the infected saliva. So here mumps affects the parotid glands and salivary glands. So here mumps is viral infection that affects a part of the body means option B, parotid gland is there, parotid salivary glands is the correct answer. Mumps usually involves pain, tenderness and swelling in one or both parotid salivary glands. Swelling usually peaks in 1 to 3 days and then subsided during the next week. Mumps is a contagious disease that is caused by a virus. Typically starts with a few days of fever, muscle ache, headache, tiredness and loss of appetite, pain and etc. So here mumps is a viral infection that affects a part of the body. Which part of the body means parotid salivary glands is the correct answer. Here in this option, option B parotid gland is there. Option B is the correct answer. Next we will move on to question number 59. Hemorrhagic viruses often kill by what mechanism? The hemorrhagic viruses often kill by which mechanism? Means we will see options first. Option A systemic shock, option B bleeding to death, option C dehydration, option D none of the above. All types of viral hemorrhagic fever characterized by fever and bleeding disorders and all can progress to high fever, shock and death in many cases. The term hemorrhagic viruses or hemorrhagic fever refers to a condition that affects many organ system of the body, damages the overall cardiovascular system, also affects through the hemorrhagic viruses. The viral hemorrhagic fever or group of disease that affects multiple organ system in the body. It may be accompanied by hemorrhage or bleeding. It may be bleeding to death. So here hemorrhagic viruses often kill by What mechanism means bleeding to death. Here option B is the correct answer. Next we will move on to question number 60. What we can found in tears? What we can found in tears? Options. Option A IgM immunoglobulin M. Option B immunoglobulin A. Option C immunoglobulin G. Option D immunoglobulin E. Our tears composed of water but also contains salt, fatty oils and over 1500 different proteins. Electrolytes in tears include sodium, which gives tears their characteristics like salty taste. Bicarbonate tears are clear liquid secreted by the lacrimal glands. The immunoglobulin A, IgA, which is a major immunoglobulin tears, is secreted as a dimer by plasma cells in the interstices between lacrimal axini. It then binds to receptor on the basolateral axis of acinar cells. It is secreted into the tear fluids. So here what is can found in tears means option B immunoglobulin A antibodies were found in the tears of all 15 subjects. So here option B is the correct answer. Next we will move on to question number 61. Which symptom exhibit during icteric phase of viral hepatitis? Which symptom we can see the during icteric phase of viral hepatitis? We will see here options. Option A shortness of breath. Option B tarry tools. Option C yellow colored sclera. Option D all of the above. What is viral hepatitis? Viral hepatitis is an infection that causes liver inflammation and damage. Several different viruses cause hepatitis including hepatitis A, hepatitis B, hepatitis C, hepatitis D and hepatitis E. Hepatitis A and E viruses typically cause acute infections. The hepatitis B, C, D viruses can cause acute and chronic infections. Patients may also report yellowish discoloration of their sclera, means icterus, and or skin. It is called as jaundice. Dark color urine and light colored stools during the icteric phase. So during the icteric phase we can see the jaundice or skin or the yellowish discoloration of their sclera. So here option C is the correct answer. Yellowish skin or yellow colored sclera develops the symptom of hepatitis A. The yellowish pigmentation arises due to the buildup of high bilirubin in the blood. Symptoms such as fever, fatigue, loss of appetite, vomiting, diarrhea and abdominal pain is the common. So here which symptom exhibit during the icteric phase of viral hepatitis means option C yellow colored sclera is the correct answer. Next we will move on to question number 62. Which of the following will cause diaper rashes? So here what is the cause for the diaper rash? We will see first options. Option A E. coli. Option B Streptococcus. Option C diphtheria. Option D none of the above. The cause strep diaper rash by bacteria is called by Streptococcus. This is also called as impetigo. It can sometimes spread into the genital area. Perianal streptococcal dermatitis is a bright red, sharply demarcated rash. Means here due to the streptococcus cause of diaper rash, it can be enters into the genital areas also. Due to this perianal streptococcal dermatitis, it will be skin will be red sharply in dermatitis also will happen. That is caused by group A beta hemolytic streptococcus. So the dermatitis streptococcal dermatitis is a bright skin is happen in the area of the diaper rash. It is caused by group A beta hemolytic streptococci. But here in this option, which of the following cause diaper rash means option B is the correct answer, streptococcus. Next we will move on to question number 63. Compound microscope was discovered by. So compound microscope was discovered by whom? We will see here options. Option A Patcher. Option B Hans Janssen. Option C Leeuwenhoek, option D, none of the above. The Jansen was the son of a spectacle maker named Hans Jansen, were the first men who developed the concept of compound microscopy. So here in this option, Hans Jansen is there in option B. So here compound microscope was discovered by Hans Jansen. The first compound microscope date to 1590s. In the year of 1590s, he only discovered, Johnson discovered compound microscope. So here in this option, option B is the correct answer. In 1519, 1590s, the Hans Johnson compound microscope was discovered. So here option B is the correct answer. Next we will move on to question number 64. Silver impregnation method. The silver impregnation method is used for demonstration of. We will see first option. Option A, Vibrios. Option B, Shigella. Option C, Spirochetes. Option D, Salmonella. Silver impregnation techniques are used to detect and enhance these organisms in histologic sections. The Golgi silver impregnation technique is a simple histological procedure that reveals complete three-dimensional neuron morphology. There are two major techniques for staining spirochetes. One consists of impregnating the spirochete with the dye or metallic ion and rendering it visible against a light background. So here the silver impregnation method is used for demonstration of spirochetes. So here in these options, option C is the correct answer. Next we will move on to question number 65. Autoclaving is usually done at. We will see first option. Option A, 115 degrees Celsius. Option B, 121 degrees Celsius. Option C, 100 degrees Celsius. Option D, 165 degrees Celsius. Autoclaving for 60 to 90 minutes at 120 degrees Celsius depending on the loading conditions. Effective autoclaving will depend on time, temperature and steam penetration. Specific temperature must be obtained to ensure the microcidal activity. The temperature must be maintained for minimal time to kill microorganisms. Autoclaving is a sterilization method. The temperature of autoclaving method for sterilization is 120 degrees Celsius. So here in these options, option B is the correct answer. Autoclaving is usually done at the 120 degrees Celsius. Next we will move on to question number 66. Streptomycin and neomycin belongs to the which class of antibiotics from the following? Streptomycin and neomycin, these all are the antibiotics. These antibiotics will comes under which class? We will see first options. Option A, macrolides. Option B, aminoglycosides. Option C, pallians. Option D, beta-lactam. So we will see one by one. Option A, microlides. What is microlides? Microlides are a class of drugs used to manage and treat various bacterial infections. For example, for the microlides, azithromycin, clarithromycin and neomycin, these all are the macrolides. We can treat through the macrolides pneumonia, sinusitis and tonsillitis. These we can treat through the macrolides. Next option C, aminoglycosides. Aminoglycosides or broad spectrum antibiotics includes gentamicin, tobramycin, streptomycin and plazomycin and neomycin. We can use it to treat severe infection as abdomen and urinary tract infection as well as bacteremia and endocarditis. So here in this question, streptomycin and neomycin belongs to the which class of antibiotics from the following means option B, aminoglycosides is the correct answer. Next we will see option C and D also briefly. Option C, pallians. Pallians are antimycotics. The antifungal agents includes nystatin, pimeracin, Used to treat oral mucosal and cutaneous infections. Next option D, beta-lactam. Includes penicillin derivatives, cephalosporin and cephalomycin used to treat bacterial infection. So here option B, aminoglycosides is the correct answer. Streptomycin neomycin will comes under the class of antibiotics, aminoglycosides. Next we will move on to question number 67. The nitrogenous base that is never found in the genetic. What we will never found in genetics according by nitrogenous base. We will see first option, option A, guanine, option B, adenine, option B, thymine, option D, uracil. Nitrogenous base not found in DNA is uracil. It is found in only RNA. In DNA, the uracil is nucleobase is replaced by thymine. Uracil is a natural base of RNA but may appear in DNA through different pathways. Not in uracil. The uracil will be in DNA like different pathways. So here nitrogenous base that is not never found in genetic is option D. Uracil is the correct answer. Uracil is not found in DNA. If it can be in RNA. So here option D is the correct answer. Next we will move on to question number 68. Copperas is used in water treatment as a. The Copperas is used in water treatment as a. We will see first option. Option A, disinfectant. Option B, indicator. Option C, coagulation. Option D, all of the above. What is Copperas? Copperas is green crystals of hydrated ferrous sulfate, especially as an industrial product. What is Copperas used for? Histologically, Copperas was used in applications such as blackening leather as a dye fixative in the industry and constitute for link. Copperas can be used as coagulation in removing the populent. Coagulation. What is coagulation? Coagulation is the action or process of liquid, especially blood, changing to a solid or semi-solid state. Copperas begins almost instantly after an injury to the endothelium lining a blood vessels. So here Copperas is used in water treatment as a coagulation. So here in this option, option C is the correct answer. Next we will move on to question number 69. Which of the following method is used for bacteriophage typing? Which of the following method can use for bacteriophage typing? We will see options first. Option A, streak method. Option B, stroke method. Option C, lawn method. Option D, stab method. In phage typing, a panel of lytic phages is inoculated on a lawn in column of the isolated under investigation. Phages which are able to set up of a lytic infection in that isolate produce a cleared zone. The method is used for bacteriophage typing is lawn method. What is lawn method? Bacterial lawn is a term used by microbiologists to describe the appearance of bacterial colonies when all the individual colonies on a petri dish agar plate merge to form field or a mat of bacteria. Bacterial lawns find use in screens for antibiotic resistant and bacterial phage titering. So here in these options, which of the following method can used for bacteriophage typing is option C. Lawn method is the correct answer. Next we will move on to question number 70. The vaccine for HSV2 is, HSV means herpes simplex virus. So here the vaccine for herpes simplex virus 2 is, we will see here option. Option A, non-existence. Option B, expensive. Option C, positive skin test. Option D, all of the above. What is herpes virus? Herpes simplex viruses have a unique 4-layered structure. A core containing the large double-stranded DNA genome is enclosed by an capsid which is composed of capsomes. The capsid is surrounded by the amorphous protein coat called as tegument. Herpes is an infection that caused by the herpes simplex virus. Although there are no currently available vaccines for herpes simplex 1 and 2, the herpes virus has more complicated DNA than most infections, has ways to go undetected by our immune system, much like many cancers cells do. Since vaccines work by stimulating the human immune system, this makes it more difficult to develop an inoculated for herpes. So currently there are no vaccines approved for prevention of herpes simplex virus infection after years of development and innovation. Non-existent and non-existent mean exactly the same thing, something that does not exist. So here the vaccine for the herpes simplex virus 2 is not existent. Option A is the correct answer here. Next we will move on to question number 71. Pentameric antibody is. The pentameric antibody is from the following. We will see here options: immunoglobulin G, immunoglobulin M, immunoglobulin A and immunoglobulin D. Here, upon recognizing antigen pentametric, immunoglobulin M is the initial antibody secreted by B cells in response to an antigen challenge. The immunoglobulin M secreted in an primary response tends to have a lower efficiency for an antigen compared to isotypes secreted on subsequent encounters or secondary response to foreign antigen, but the pentameric antibody is the primary and initial antibody. So here the pentameric antibody is a immunoglobulin M. So here in this option B is the correct answer. Next we will move on to question number 72. Which of the following structure is the smallest? From the following option, which is smallest structure? We will see first options. Option A, virion. Option B, mitochondrion. Option C, hydrogen atom. Option D, none of the above. The atom is the smallest and most fundamental unit of matter. It is consist of a nucleus surrounded by electrons. Atoms combine to form molecules, which are chemical structures consisting of at least two atoms held together by a chemical bond. Hydrogen atom is an atom of the chemical element hydrogen. Hydrogen atom is the smallest and simplest of all atoms. It is consists of a single proton and single electron. So here, which of the following structure is smallest means hydrogen atom is the smallest structure in the body. So here option C is the correct answer. Next we will move on to question number 73. Bacterial structure involved in respiration. Which bacterial structure will involved in the respiration process? We will see here options. First option A, pili. Option B, mesosome. Option C, ribosome. Option D, bacterium. The site of aerobic respiration in the bacterial cell walls is mesosome. Cellular respiration is an energy generating process that occurs in the plasma membrane of bacteria. Mesosomes are folded impregnation in the plasma membrane of bacteria. Mesosomes perform various functions such as various respiratory enzymes are present in mesosome. Mesosome helps in cellular respiration. So here bacterial structure involved in respiration from the following is mesosome. Option B is the correct answer. Next we will move on to question number 74. In which condition the signet ring is seen? In which condition we can see the signet ring? We will see options. First option A, measles. Option B, malaria. Option C, polio. Option D, all of the above. Malaria is caused by unicellular eukaryotic parasites from the Plasmodium genus and ring form tropojets of Plasmodium falciparum, often thin and delicate, measuring on average 1/5 the diameter of the red blood cells. The Plasmodium vivax is characterized by thicker single signet rings. All stages of the parasite are seen in the peripheral circulation. during the malaria. So here in which condition we can see the signet ring is seen in the options, means option B, malaria is the correct answer. During the malaria we can see the to the red blood cells plasmodium vivax is caused by the thicker signet ring in the blood. So here option B is the correct answer. Next we will move on to question number 75. Which toxin of Staphylococci is responsible for the manifestation of poisoning? Which toxin of Staphylococci is responsible for the poison manifestation? We will see first option. Option A, leucocidin. Option B, enterotoxin. Option C, hemolysis. Option D, exotoxin. Staphylococci food poisoning is an intoxication that results from the consumption of foods containing sufficient amounts of one or more performed enterotoxins. So the toxin of Staphylococci is responsible for the manifestation of poisoning is enterotoxins. So here option B is the correct answer. The signs and symptoms for the symptoms of manifestation of poisoning is nausea or vomitings, abdominal cramping, and with or without diarrhea. So here in these options, option B, enterotoxin is the correct answer. Next we will move on to question number 76. What is the causative agent? What is the causative agent of plague? Means what is the cause of agent for the plague? We will see here option. Option A, Mycobacterium leprae. Option B, Yersinia pestis. Option C, mosquito. Option D, Mycobacterium tuberculosis. Here option A, Mycobacterium leprae means related to the leprosy. Next option D, Mycobacterium tuberculosis means it is nothing but TB, tuberculosis or Koch's disease. Here option B or C we will see. The plague is an infectious disease that affects from the animals. So here option C, mosquito is not comes under here. So option B, Yersinia pestis is the correct answer. Plague is an infectious disease that affects from the animals and humans. It is caused by the bacterium. The bacterium was Yersinia pestis. Yersinia pestis is the correct answer. Yersinia pestis is a gram-negative bacteria. Plague has most famously been called the Black Death. Because why we will call Black Death? Plague will also called as Black Death because it can cause skin sores that form of black scabs. Plague spreads mostly by fleas on rodents and other animals. For example, for the condition is zoonotic disease. Here the causative agent of plague is option B, Yersinia pestis is the correct answer. Next we will move on to question number 77. A client had an indwelling urinary catheter for 48 hours in perioperative period. On revisit after 1 week, he has a urinary tract infection. It is termed as, means the client had a indwelling urinary catheter for 48 hours in the perioperative period and revisit he had a urinary tract infection after 1 week. This is called as, we will see options. Option A, recurrent urinary tract infection. Option B, common urinary tract infection. Option C, iatrogenic urinary tract infection. Option D, none of the above. A client had indwelling urinary catheter for 48 hours in perioperative period. On revisit after 1 week, he had a urinary tract infection. This is termed as iatrogenic injury to the urinary tract. The iatrogenic injury to the urinary tract including the kidneys, uterus, bladder and urethra is a possible complication of surgical procedure. Means it is a possible complication of post procedure performed in the retroperitoneal abdominal space or in around the pelvis. So here the client had an indwelling urinary catheter for 48 hours perioperative period and revisit after 1 week he had a urinary tract infection is iatrogenic urinary tract infection. Option C is the correct answer here. Next we will move on to question number 78. Cytokinase are chemical messengers which the cytokinase are chemical messengers which are, we will see first option A, kill microorganism, option B, promote inflammation, option C, severe pain, option D, produce antibodies. The cytokines are the primary chemical messengers involved in both acute or chronic inflammation. Chemical messengers also called as cytokinase. The cytokines plays an important role during the body's initial response to infection. So here the cytokines are chemical messengers which can promote inflammation. So here in this option B is the correct answer. Cytokines affect the growth of blood cells and other cells that help the body immune and inflammation responses. So here promote the inflammation is the correct answer, option B. Cytokines are chemical messenger for the promote inflammation. Next we will move on to question number 79. Which infection is diagnosed with the Paul-bunnell test? So here the Paul-bunnell test will diagnose the which condition? We will see first options. Option A, arthritis. Option B, rheumatic arthritis. Option C, infectious mononucleosis. Option D, all of the above. The Paul-bunnell test is used to screen for infectious diseases related to the infectious mononucleosis, which is seen most commonly in adolescents and young adults. Infectious mononucleosis is caused by the Epstein-Barr virus, EBV, Epstein-Barr virus. The infectious mononucleosis, also called as mono, often called as mono or kissing disease, a infection with Epstein-Barr virus. The infectious mononucleosis is transmitted by saliva from mouth who are infected. The signs and symptoms for the mononucleosis is swollen lymph nodes and swollen lymph glands, fever, sore throat and extreme fatigue. These all are the signs and symptoms for the condition of infectious mononucleosis. Symptoms can be take between 4 to 6 weeks to appear and usually do not last beyond 4 months. Till 4 months the symptoms will not disappear. So here the which infection is diagnosed with the Paul-bunnell test is option C, infectious mononucleosis is the correct answer. Here option C. Next we will move on to question number 80. The 80th question is the last question in this today's class. We will see the optimum temperature for the growth of most pathogenic bacteria. The optimum temperature for the growth of pathogenic bacteria. We will see first options. Option A, 37 degrees Celsius. Option B, 30 degrees Celsius. Option C, 28 degrees Celsius. Option D, 20 degrees Celsius. Human pathogens generally multiply best at temperature similar to those of the human host. The temperature it will be 35 degrees Celsius to 37 degrees Celsius is the normal. But this is because pathogens are mesophils, which means they function best at the human body temperature of 37 degrees Celsius. Yet this temperature their enzymes are very active and they are not denatured by the high temperatures. So the optimum temperature for the growth of most pathogenic bacteria is option A, 37 degrees Celsius is the correct answer. In this temperature pathogens are mesophils, which means they function best at the human body and temperature their enzymes are very active. So here option A is the correct answer, 37 degrees Celsius. 8th question is the last question in this part 3 video related to the microbiology subject. We will continue from the 81 question to 100 question, means 20 questions we will continue in further video part 4. Next video we will continue. Thank you so much for watching my video. Once again thank you so much for everyone. Anybody watching first time my channel, please subscribe my channel, click on the bell icon for further notification. Please like the video and share the video to your friends and share your opinion in comment section. Stay tuned friends. Thank you. Keep watching.